Hello, everyone, and welcome to today's webinar, Analytical and Clinical Validation Requirements for Next-Generation Sequencing Laboratory-Developed Tests. I am Michelle Ashton of Labroots, and I'll be your moderator for today's event. Today's educational web seminar is presented by Labroots and brought to you by Thermo Fisher Scientific. To learn more about our sponsor, please visit www.thermofisher.com. Let's get started. I would like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want at any time you want during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. If you have trouble seeing or hearing the presentation, click on the Support tab found at the top right of the presentation window, or report your problem by clicking on the Ask a Question box located on the far left of your screen. This presentation is educational and thus offers continuing education credits. Please click on the Continuing Education Credits tab located at the top right of the presentation window and follow the process to obtain your credits. I now present today's speaker on behalf of the College of American Pathologists. Dr. Ryan Robetorye, Director, Clinical Laboratories at the Mayo Clinic in Arizona. For a complete biography on our speaker, please visit the Biography tab at the top of your screen. Dr. Robetorye will now begin his presentation.
Hi, everyone. As just mentioned, I serve as the medical director of our clinical laboratories here at the Mayo Clinic in Phoenix, Arizona, and I did not have any relevant financial relationships to disclose other than my employment at the Mayo Clinic. Since the College of American Pathologists, or the CAP as we commonly call it, is the major accrediting body for clinical laboratories that perform molecular diagnostics testing in the United States and around the world, my discussion today will describe the requirements that laboratories need to fulfill in order to validate next-generation sequencing-based testing from the perspective of the CAP molecular pathology and all common checklists. So today we'll discuss the concept of laboratory-developed tests, or LDTs as they're known, which also requires some basic knowledge of the FDA and what it means to be FDA-approved and FDA-cleared. Today we'll also discuss the required requirements of an LDT validation, next-generation sequencing-based testing validation components and requirements, as well as the concept of a next-generation sequencing-based validation summary, and then we'll also have some time for questions at the end of the presentation. So now for just some basic definitions. What is the FDA? For laboratories that are subject to U.S. regulation, the FDA refers to the U.S. Food and Drug Administration regulatory body that has the authority to regulate in vitro diagnostic products such as testing kits, reagents, instruments, and testing systems. FDA-approved basically means that the agency has determined that the benefits of the product outweigh the known risks for the intended use of the product. And the concept of FDA-cleared is basically when a manufacturer can demonstrate that their product is substantially equivalent to another similar legally marketed device that already has FDA approval. So with very few exceptions today, most next-generation sequencing tests are LDTs. So what exactly is an LDT? An LDT is simply a test that's used for patient management that has the following features. The test must be performed by the clinical laboratory in which the test was developed, either wholly or in part, and the test must not be FDA approved or FDA cleared. So what are the validation requirements for an LDT? Each laboratory must establish the analytical performance parameters for each LDT is a part of their assay validation process, and the analytical performance parameters must include accuracy, precision, analytical sensitivity or the lower limit of detection, analytical specificity or interferences, reportable range, and reference intervals. So over the next 2 slides, we'll briefly discuss the analytical performance parameters. Accuracy is basically determined when there is a comparison of results to a definitive reference method or an established comparative method in order to establish the relative trueness of results. And this is accomplished basically using reference materials, clinical patient samples, or other representative sample types. Precision or reproducibility is the process in which the laboratory demonstrates that test results are consistent when they are repeated by different individuals over time. And then the concept of analytical sensitivity or lower limit of detection is basically each laboratory has to demonstrate a lower limit of detection for each of the LDTs that they run in their clinical lab. The next category is analytical specificity or interfering substances. Each laboratory must demonstrate that their testing produces accurate results even in the presence of possible interfering substances that might expect it to be present. And each laboratory must also establish a range of values that will be reported for each of their assays. And this is known as the reportable range. The analytical measurement range, or AMR, is basically the range of values that an assay can actually measure without any dilution, concentration, or other pretreatment that would not be a part of the usual assay procedure. And then the final analytical performance parameter is the reference interval, which is basically the range of results that are expected in the normal population. Every LDT must also have an associated validation summary document that reviews all the analytical and clinical performance parameters of the assay. So we just discussed the analytical performance characteristics, the concept of test accuracy, precision, analytical sensitivity, analytical specificity, the reportable range, and reference interval. But the clinical performance or clinical validity characteristics of the assay must also be summarized in the validation summary. So the concept of clinical sensitivity, or the ability of a test to identify patients with a disease, must be summarized in the document. Clinical specificity, or the ability of a test to identify patients without a disease, must also be discussed. And the concept of clinical utility, which is basically a measure of the relevance and usefulness of the testing for individual patients, must also be a part of the summary. However, laboratories are also allowed to cite relevant scientific literature in their validation summary if the clinical performance parameters can't be established within their own laboratories. So now we're going to transition from the discussion of LDTs to next generation sequencing validation requirements. And for this section, we're going to use the relevant CAP molecular pathology and all common checklist items to illustrate this process. So the first item for discussion from the molecular pathology checklist involves specimen types and requires that all validation studies have an adequate number of representative clinical samples for each of the specimen types that are expected to be utilized by the assay. So, for example, clinical specimens such as blood, fresh or frozen tissue, saliva, paraffin-embedded tissue, prenatal specimens, and buccal swabs, etc., should be utilized as a part of the validation study. The associated note for this requirement also states that validation studies can be supplemented by additional reference materials, such as previously characterized cell lines or cell lines, for example, that might have spiked in nucleic acids of known concentration. The next requirement from the All Common checklist requires that all next-generation sequencing LDTs include an appropriate number of samples. The associated checklist note for this particular requirement requires a minimum of 20 samples for the validation, and also states that for tests like next-generation sequencing that test multiple analytes, the validation of all possible variants that might be detected is not necessary, and a methods-based validation is acceptable. And we'll discuss this concept a little further later in the presentation. The next item from the molecular pathology checklist that's applicable to next-generation sequencing requires that each validation study include a sufficient number of previously characterized samples in order to assure the accuracy of the testing. And the associated note for this particular requirement also states that pathogenetic variants representative of the type that will be detected by the assay should also be a part of the validation in order to confirm the accuracy of the testing method. Another item from the molecular pathology checklist requires that the results of each validation study include a sufficient number of samples with repeated analyses in order to provide a high degree of assurance of the test's precision or reproducibility. And the associated note for this particular checklist item requires that laboratories must be able to document that their testing will return the same result regardless of minor changes in testing conditions that might result in random errors, such as using different technologies, different instruments, reagent lots, or testing days. The next item from the molecular pathology checklist applicable to next-generation sequencing requires that laboratories also include a sufficient number of samples in their validation study in order to confirm or establish the reportable range for the test. So for next-generation sequencing-based testing, the reportable range includes all the regions of the genome or the transcriptome for which the assay can accurately produce sequence information. So, for example, for whole genome testing, the reportable range would be all the bases of the genome. Or it could be the exome for exome testing. It might also be a list of genes or gene regions for gene panel sequencing, the sequence of an individual gene, only selected exons, or maybe the whole transcriptome for transcriptome RNA-seq, or maybe even only a subset of transcripts for targeted RNA-seq-based testing. So the next item from the molecular pathology checklist applicable to next-generation sequencing requires the establishing of a testing reference interval. And since the reference interval is the range of expected results in the normal population, the relevant reference interval for next-generation sequencing-based testing is simply comprised of the most recent human reference genome assembly, which right now, of course, is the Genome Reference Consortium Human Build 38 Patch Release 13, which was last updated in March of 2019. Another applicable molecular pathology checklist item requires the establishment of the test's analytical sensitivity or lower limit of detection. And as detailed in the associated note for this checklist item, this requirement refers to the ability of a test to consistently detect a minor allele or variant in a background of assay-relevant biological matrices such as pathogens that might be present, rare variants, the presence of chimerism, mosaicism, or even tumor-normal mixtures. Another molecular pathology checklist item requires that laboratories performing next-generation sequencing-based testing to establish a lower limit of detection for mixed populations. Since we know that the lower limit of detection can vary for different variant types, such as single nucleotide variants, insertions and deletions, copy number variants, and other structural variants such as translocations and inversions, the limit of detection should also be determined for each variant type that the assay is actually intended to detect using previously characterized samples in which the variant allele fraction is already known. The next item from the molecular pathology checklist applicable to next-generation sequencing-based testing is a requirement to establish the analytical specificity for the testing. And as previously noted, analytical specificity is the ability of a test to correctly identify or quantify an entity in the presence of interfering or cross-reacting substances that might be expected to be present. We know that interfering or cross-reactive substances might be specimen or tissue type dependent, such as the hemoglobin that's present in blood specimens, melanin that might be associated with melanoma samples, or other substances that may be associated with some samples such as lipids, mucin, heavy metals in fixation methods, or even contaminating cells. Specimen carryover can also be a problem with next-generation sequencing-based tests, especially those that are designed to detect variants with low allele frequencies. An item from the all-common checklist applicable to next-generation sequencing-based testing requires that any clinical claims made by the laboratory must be validated for all laboratory-developed tests. Some examples of clinical claims might include statements about the diagnostic sensitivity and specificity of the testing, the ability to predict risk of disease. It might be statements relative to the clinical usefulness of a test or even its cost effectiveness. This checklist item also states that the clinical laboratory must perform a clinical validation study to support any of the clinical claims that they make. And this validation requires a minimum of 20 samples. Another item from the molecular pathology checklist requires that the clinical performance characteristics of each assay are determined and recorded either using literature citations or a summary of internal study results. The clinical performance characteristics of a test basically relate to the diagnostic sensitivity and specificity of the testing, positive and negative predictive values in the target population for the testing, and also the clinical utility of the testing. The next item from the molecular pathology checklist regarding next-generation sequencing wet bench validation requires that all next-generation sequencing laboratories validate their analytical wet bench processes and then revalidate these processes when modifications are made. The analytical wet bench process and bioinformatics processes for any given test will likely occur within a single laboratory, but in today's world we know that a combination of primary and referral laboratories may also be present, with the wet bench process, for example, performed in the primary laboratory and then the laboratory referring its bioinformatics analysis to another company. However, even if these two processes occur in different laboratories, the validation of both the wet bench and bioinformatics processes must be integrated to ensure that there is acceptable beginning to end performance for the whole pipeline. Analytical wet bench validations must also include a methods-based validation to establish the test's general performance for detection of the sequence variant types that the assay is designed to identify. And the validation must also establish metrics and quality control parameters that will be used to assess the wet bench process performance. It's important to note that it's the primary responsibility of the laboratory director to review and approve all validations that are relevant to the intended tests for the processes performed in their laboratory, as well as for any processes that might be performed in referral laboratories, if applicable. Another important item from the molecular pathology checklist applicable to next-generation sequencing testing requires that all next-generation sequencing laboratories also validate their analytical bioinformatics process and then revalidate these processes when any modifications are made. The analytical wet bench process and bioinformatics processes for a test might occur again within a single laboratory, as is done in most cases, or in a combination of primary and referral laboratories. However, the validation of both of these processes, the wet bench and bioinformatics processes, must be integrated to ensure beginning to end test performance. A methods-based approach can also be used for the analytical bioinformatics process validation, using a sufficiently high number of variants that the test is designed to detect, such as single nucleotide variants, insertions and deletions, copy number variants, as well as other structural variants such as translocations and inversions. As with the wet bench process, metrics and quality control parameters must also be established to assess the bioinformatics process performance during the validation. And again, it's the primary responsibility of the laboratory director to review and approve all of these bioinformatics process validations and also bioinformatics processes that might be performed in possible referral laboratories. This next generation sequencing related item from the molecular pathology checklist requires that there must be an actual validation summary document that addresses all the analytical and clinical performance parameters of the testing. And of course, the laboratory director must review and approve this validation summary prior to starting any clinical testing, and that's a very important point that is overlooked by some laboratories, that any testing that they perform must be approved by their laboratory director prior to any clinical testing being performed by the laboratory. This slide just shows some of the detailed steps that should be included in a good validation summary. So you need a detailed step-by-step description of the assay and how it will be implemented. Clinical indications and the intended use of the assay should also be included in this summary. The name of the assay and any instruments that are used. All methods and reagents required for the assay should be a part of this summary. The FDA status or LDT designation of the testing. A very important part of the validation is all the software scripts and software version numbers in the bioinformatics pipeline. And a good practice for any laboratory using a pipeline is to name each version of their pipeline from beginning to end so that it could be reproduced if needed. All specimen types and numbers of specimens should be listed in the validation summary. All the analytical performance and clinical performance characteristics should be present. Any quality control and quality assurance metrics to ensure that the testing is being done properly should be in the validation summary. And there should be a description also of any required proficiency testing, which could come from the College of American Pathologists, or it might be an alternative proficiency testing method with a detailed description and acceptable criteria for the testing. Any relevant safety issues related to the testing should be listed. You should document notification of the CAP if you're an accredited CAP laboratory that you're initiating a new test. And good practice also requires a statement like I've listed here. This validation study has been reviewed and the performance of the method is considered acceptable for patient testing. And there should be a section in the validation summary for the laboratory's director's signature with date, and also the date that the actual patient testing began, which of course should be sometime either the day that the laboratory director signs it or sometime after that. So what are the specimen types that should be used for a next-generation sequencing validation? The molecular checklist item MOL.3113015 requires that the validation must include representative sample types that are expected to be tested by the assay, including such specimen types such as peripheral blood, plasma, bone marrow specimens, saliva specimens, other body fluids, fresh tissue or fresh frozen tissue, formalin-fixed, paraffin-embedded tissue samples, buccal swabs, or perhaps even prenatal specimens. Previously characterized clinical samples with each type of pathogenic alteration that the assay is intended to detect should also be used. So if your assay is designed to detect single nucleotide variants, for example, examples of those samples should be included. And if the assay is designed to detect insertions, deletions, structural variants, or copy number variants, there should be examples in your validation of all those type variants. Other validation materials that could also be used include proficiency testing materials, and a very useful category that we use actively in my laboratory for testing validation is in silico generated data sets for bioinformatics analysis. And these are basically sequence files that were obtained from next generation sequencing analysis of well-characterized reference specimen that have then been manipulated by computerized algorithms in order to introduce sequence variants at various allele frequencies for subsequent analysis and detection. So these types of data sets are extremely useful for testing the bioinformatics portion of your next generation sequencing pipeline. Commercially available reference materials may also be used for next generation sequencing validations, and there are many of these available. I didn't list any specific companies, but they're pretty easy to find, and we've used many of these in my own laboratory for validation purposes. You can use the very well characterized human genome reference standard, the NIST cell line NA12878. There are many commercially available reference genomic DNA standards that have different types of variants at known variant frequencies, including very low allele frequencies to help you test your lower limit of detection in your assay. And there are also a number of commercially available reference FFPE specimen standards with known allele frequencies and variant types that you can use if your assay is designed to detect variants in FFPE tissues. So a very common question that I get and the CAP gets is, how many specimens should I use for my next generation sequencing validation? The all-common checklist item COM.40350 requires a minimum of 20 samples for any test validation. And there are really no other number limitations stated in the checklist requirements. And this leaves the actual number of specimens used for the validation at the discretion of the laboratory director, and the validation summary should justify whatever number of specimens are chosen for the validation. And of course, the validation should also include previously characterized samples of the specimen type that are intended to be used for the assay, as well as specimens with pathogenic alterations that are intended to be detected by the assay. A recent publication from Jennings et al. In the Journal of Molecular Diagnostics, I have the reference listed on the slide, the bottom part of the slide, recommends using a minimum of 59 samples for validation of a next-generation sequencing oncology panel. And today, I think this is actually a pretty reasonable recommendation. In a recent validation that we performed of a 425 gene panel, we used over 100 validation samples, and I think this is really reasonable today because there are so many clinical specimens available from other laboratories performing next-generation sequencing that you can obtain a number of commercially available reference standards, as well as I mentioned, the very useful NIST cell line NA12878. This slide contains a number of next-generation sequencing validation articles from a recent issue of the Archives of Pathology and Laboratory Medicine, and all these articles contain useful information regarding next-generation sequencing testing validation, and you can use those as you see fit. Another good recent manuscript from Dr. Avni Santani and her colleagues was published in the May 2019 issue of the Journal of Molecular Diagnostics, as noted here on the slide. And this manuscript contains many practical step-by-step guidance instructions for next-generation sequencing-based testing validations. And finally, additional helpful next-generation sequencing laboratory worksheets are also available from the CAP public website that discusses various topics such as test content design, test optimization, test validation, quality management for the laboratory, as well as bioinformatics processes in the lab. So that is my last slide, and I'll be happy to entertain any questions if there are some. Thank you.
Thank you, Dr. Robetorye, for your informative presentation. We will now start the Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the Ask the Question box located on the far left of your screen. We'll answer as many questions as we have time for. Okay, let's get started. Our first question is a revalidation of our whole exome sequencing bioinformatics pipeline required if our bioinformatics pipeline is updated to a new version?
The short answer is yes. So anytime any changes are made to either the wet bench process or the bioinformatics pipeline, the assay should be revalidated from beginning to end to ensure that testing accuracy is maintained, even in the context of the changes that were made to the testing methodology. So that's a very important requirement, and any good laboratory should be revalidating their process anytime any changes are made, either to the wet bench analytical process or the bioinformatics pipeline.
Great, thank you. Okay, next question. Must our NGS validation studies include all possible mutations detected by our analysis of gene regions that we routinely analyze and report out?
Well, of course, it's not realistic and most likely not even possible for most generation sequencing validation studies to include all the possible genetic alterations that might be expected to be detected by the testing. So, in this context, as mentioned before in the presentation, a methods-based validation approach is completely acceptable as long as it includes the types of specimens that are expected to be tested in the assay, as well as the categories of variant types that are expected to be tested by the assay.
Thank you again, Dr. Robetorye, for your time today and your important research. We would also like to thank Labroots and our sponsor, Thermo Fisher Scientific, for underwriting today's educational webcast. Before we go, I'd like to thank the audience for joining us today and for their questions. Any questions we did not have time for today and those submitted during the on-demand period will be addressed by the speaker via the contact information you provided at the time of registration. This webcast can be viewed on demand. Labroots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's event. Until next time, goodbye.